Hi, my name is Sonakshi and I am a high school student living in Penang Island, Malaysia. This is my first video in the series of biology experiments. This series is especially for people who have a curious mind and want to explore the world of biology. So, let's begin. My today's video is about plant epidermal cells, onion peel cells. I am going to make a specimen slide about onion peel cells and view them under a compound microscope. Now I am back with all of the apparatus and material we are going to use. I have an onion, a piece of onion here with me to extract the peel, a compound microscope, the glass slide, the cover slip, the forceps, and also the pigment. I am using methylene blue pigment. You can use saffron, iodine, or crystalline purple. You can also use any other pigment you have or you like. Here I am going to extract a piece of onion peel from the onion, and here I have, yes, the peel you extract may not be equal, so you can just cut it out or place it the way it is already is here. When you are done with it, you will need a drop of water to make sure this peel doesn't dry out, the cells in the peel doesn't dry out. So here you have this dropper. Using this dropper, I am going to add drop of water on the onion peel cells. After that, I am going to add methylene blue solution. And now I am going to place the cover slip at the angle of 45 degrees using a forceps. I am going to wipe away the extra liquid. Now this looks good. What you can do next is to just leave it for 30 seconds to wait for the peel to absorb the pigment. Now while we are waiting, let us learn what onions are exactly. Many of you will tell me that it's a root. Some smart one of you will tell me it's a stem. And some of you would already know the answer what it is. But an onion is not exactly a stem nor it's a root. It is a tunicated bulb which are used to store food. This tunicated bulb is actually a modified stem. Yes, it is a stem but a modified one. The leaves, the fleshy leaves of an onion are actually the ones which store the food. So here we know a lot about onion. Now onion are also present underground. You may have seen it. So they don't have chloroplast in it, as there's no use of chloroplast under the ground, as you cannot access sunlight. Now I think 30 seconds are done. Here I have my slide and I think the pigment is absorbed very well. So next we are going to see it under a microscope. Hey, here I am with the compound microscope. At the top we have the ocular lenses or the eyepiece. Then we have the head and the arm. Here is the arm. Attaching to the arm we have two knobs. The first one, coarse knob, and the second one, the fine knob. We call it the coarse focus and the fine focus. Down here after the arm we have the nose piece. Attaching to the nose piece we have the objective lens. Then we have the stage. On the stage we have the stage clips. We use the stage to place the slide on, and the stage clips to make sure the slide stays and be fixed on the position. Then at the very bottom we have the mirror to pass the light so that we can see the slide or the cells or microorganisms on the slides. With the mirror, I use a lamp or any artificial light source to reflect the light, or you can just use the direct light from the sun. Now I have my slide. I labeled my slide so that I can know which the slide is about. I labeled it the onion peel cells blue pigment. Now I'm going to place my slide on the stage and fix them by the stage clips. Here they are. I'm going to set my lenses. Here I have the 30x lens, the 2x lens and the 10x lens. So 30 multiplied by 2 multiplied by 10 equals to 600x. I'm going to make sure the mirror is reflecting the light on the right path, and here it's already. So now we are going to look at the cells from under the compound microscope. Interesting? We will find out. So here we are under the compound microscope looking at onion peel cells. Onion peel cells are epidermal cells because they provide a protective layer to the fleshy modified layers of the onion bulb. And as we all know, onion peel cells are plant cells because they have a cell wall, cell membrane, vacuole, cytoplasm and nucleus, but they don't have a chloroplast. Now we are going to study and learn about these structures in detail. So here is the cell wall of the onion peel cells. As you can see, the outer layer covering each cell over this onion peel specimen slide. So the structure of this cell wall is a rigid layer surrounding the cell, and its function is to support, protect and provide shape to the cell. Now here we have the cell membrane just right inside the cell wall. It is quite hard to see the cell membrane, but if you look closely you can see the cell membrane. The structure of the cell membrane is just an inside layer of the cell wall, and its function is to control substances that go in and out of the cell. Now these are the cytoplasms. You can see cytoplasms in each cells. Cytoplasm is clear, thick, gel-like fluid, and its function is to hold organelles in place. Now we have the nucleus in each cell. This round structure you can see, they are the nucleus. Nucleus has a large spherical structure. Nucleus is the control center of the cell, which holds the genetic material, the DNA. The nucleus is also known as the brain of the cell because it regulates development and metabolism. Now here we have the vacuoles. You can see the patches, white patches. Yes, they are the vacuoles. The structure of the vacuole is one large water-filled sac, and it is a storage area for cells for food, material and waste. So here it was all about these onion peel cells. In today's video, we learned how to create specimen slide for onion peel cells. We also learned about compound microscope and some basic organelles of a cell. Now it's trivia time. Did you know the man who created high magnifying lenses for compound microscope wasn't actually a scientist, but a draper, a man who deals in clothes? He created these lenses to check the quality of the threads of his clothes, but he ended up observing cells. Eventually, he started observing microorganisms, and he found out that they are living and alive. Today, this man is known as the father of microbiology. Do you know his name? Antony van Leeuwenhoek. Surprising, right? Now, if you liked my today's video, please like, share and subscribe my video. Thank you.